Good morning, biology, and welcome back. Today we're going to be talking about the fundamentals of genetics. We're talking about how we get DNA from into chromosomes, and then actually turn that into genes and genetics, and that's kind of going to be the focus of the next couple weeks. Today we're going to talk about this guy, Gregor Mendel. We're going to talk about his legacy. He's kind of the father of genetics, as we call him. There's a little controversy on that, but it's all right. For what we're going to talk about today, we're going to talk about him, his kind of accomplishments, how to do some basic genetics, and then we're going to talk about Punnett Squares as well, everyone's favorite part of biology. All right, so first up, who was Gregor Mendel? Gregor Mendel was a monk. He worked in an Austrian monastery, and in 1815 he entered the University of Vienna to study science and to study mathematics. His math courses, and this is kind of important, involved the brand new field of statistics, and that's where a lot of his findings for genetics come from, is actually from statistics. This was very useful on his research for heredity. His ability to use a large quantity of numbers and make a meaningful story out of them is kind of what sparked and kind of drove this force behind Mendelian genetics. Now, heredity, to define that, is the transmission of characteristics from parent to offspring. So when someone says you look a lot like your dad or you look like a lot like your mom, that's heredity. You've got those characteristics from your parents. They have been transferred to you. So Mendel, he worked on pea plants in the monastery, and he was looking at 7 different characteristics of pea plants, and trying to grow the perfect pea. But the traits that he looked at, and you can think of a trait as just any genetically determined variant of a characteristic. So for seed shape, it could be round or wrinkled. For color, it could be yellow or green. These are all different traits that these seeds have. Flower could be purple or white. So he looked at lots of traits: pod color, plant height, flower color, seed texture, the flower position, whether or not the pod had a color or shape of the color. So there was a lot of things that he was looking at. Like I said, keeping a lot of numbers, moving around. It's a good thing he had statistics. All right, so Mendel would collect the peas from his plant. He would record each pea's traits and the seeds that they got from it. And then he would use those to grow the next generation. But what happened is the results were not always what he expected. Sometimes he got short plants from tall seeds, and sometimes he got white flowers from purple parents. And so he had to explain, how does this happen? How does he get this white trait from these purple parents? It doesn't appear that they have that trait. So let's talk about Mendel's method. Mendel very carefully controlled how the plants were pollinated. And pollination is when the pollen grains here inside of this little anther here in the male reproductive part of the female plant are transferred to the female part, the stigma. And there's ovaries inside of here, same thing as we kind of think of ovaries inside of humans. The anther is the male. It's these kind of little purple parts on this flower, and then the stigma is the big green part and is the female reproductive part. Now, for some flowering plants, they have 2 ways to pollinate. They can either self-pollinate or cross-pollinate. And self-pollinate is when the pollen from one of the anthers on that plant, or of one flower, is then sent to the stigma on the same flower. So this is self-pollination. This pollen is going from the same flower from the anther of the same flower to the stigma of the same flower. And in cross-pollination, this is occurring when there's 2 flowers and 2 plants. So the pollen from this anther is going all the way to the stigma of a different plant during cross-pollination. So normally pea plants are self-pollinators, but Mendel would use brushes, and he would manually pollinate the pea plants, and he'd cut all the flowers off except for a couple so that he could control the pollen. And he would take the pollen from one and he would cross-pollinate to another one. So kind of a cool experiment. In the beginning, Mendel's experiments, he was looking at only one trait at a time. And what he would do is he would take plants that were true breeding. And these were plants that when they self-pollinated with each other, they always produced offspring with the same trait. So, example, if you had a plant with yellow pods and it self-crossed, it self-pollinated, it would only produce offspring with yellow pods. You wouldn't ever get a green pod. And same thing, if you had a true breeding green pod, it would only ever produce green pods. It wouldn't ever produce yellow pods. Mendel eventually had 14 true breeding plants, one for each of the 7 traits. So one wrinkled, one smooth, one yellow, one green. He referred to these guys as the P generation, or the parental generation. So in this one here, this is the true breeding purple flower, and this is the true breeding white flower. He then bred these two together to see what would happen. What would he get as the results? An example we're going to look at is purple flowers, and then mixing them with white flowers. So in the F1 generation, which stands for the Latin for filial, which is Latin for son of or a daughter, he got this first filial generation, and you can see here all of them have purple flowers. All 4 of these have purple flowers. This white trait kind of disappeared somewhere in here. So then what he did is he took 2 of these guys, these 2 let's say, and he's going to breed them together. He's going to take 2 of these F1s and breed them together and see what the offspring look like. And what he got was the F2 generation down here, the second filial generation. And he then recorded the data for all these crosses and all 14 traits. But you can see here there's this really cool 3-to-1 ratio that we're going to talk about in the next kind of slide, because this white trait's now back. It skipped a generation and came back. And if you hear people say, oh, it skips a generation, they're talking about this type of trait. So Mendel's results. When a true breeding green was crossed with a true breeding yellow, the F1 was always 4 out of 4 green, or in our example, 4 out of 4 purple flowers. The F2 were 3 out of 4 green and 1 out of 4 yellow. And this kind of 3-to-1 ratio that we see is very, very typical of Mendelian genetics. So what Mendel concluded is that the factors were inherited and that each trait was compared by a pair of factors. Now, the factors could either be recessive or dominant. So when the experiments in the P generation were crossed, one trait would always disappear. The one that disappeared is called the recessive factor, and the one that stayed is called the dominant factor. The dominant factor always covers the other factor, and the recessive is always hid by the other factor. So if you have a mom and a dad, and the dad is dominant brown eyes, and the mom is blue-eyed, the child will have brown eyes. The brown is dominant, it hides this blue recessive trait. Now, remember that we get these traits through meiosis, and we got to talk about a couple laws. Again, we got to talk about the law of segregation first. Each gamete inside of when the meiosis happens has its one copy of a factor. So we start out with 2 chromosomes. Dad chromosome gets split to one end, mom chromosome gets split to the other, and then it gets split again to form these 4 different factors here. When we combine offspring, we're combining one from dad and one from mom to make a zygote. So the law of segregation says that pairs of factors segregate or separate during the formation of gametes. So chromosome, let's say this is chromosome 3. Each gamete is only getting one copy of chromosome 3, whether it's from dad's original side or mom's original side. The second law we need to talk about is the law of independent assortment. And in this one, different traits are coded for by different factors. So a green pod can be wrinkled, or it can be a green pod and be round. So these guys can be, each of these factors differently separate out. So remember that homologous chromosomes randomly separate during anaphase 1 of meiosis. And so the law of independent assortment simply states that factors separate independently of one another during gamete formation. So if you look at these 4 daughter cells, none of them have the same genetics as each other. They are all completely independent. They all have different traits. They all have different kind of things that we're going to expect to see. So there's some support for Mendel's conclusions. We've recently, starting in the 1900s, we started molecular genetics, which is the study of chromosomes and their genes. And a gene is a segment of DNA on a chromosome that controls a particular hereditary trait of Mendel's. And then, so this right here, this dark blue region is a gene. And then the allele is the different forms of the gene. So in this one, if there's a little b and a big B, they're 2 different forms of the same genes. So we now call Mendel's factors just simply alleles. He was right on track. He had the right idea, but we now call them alleles instead of traits. All right, let's talk about how we write alleles, an allele notation. Alleles are generally represented by numbers, things like A or P or R or S or T or things like that. Dominant alleles get the capital letter, and recessive alleles get a lowercase letter. Alleles coding for the same gene are usually the same letter. So if you have, let's say, a gene that you want to call Y, the dominant will be big Y, the recessive will be little y. All right, going to jump right into, this is the end of 8.1. We're going to jump right into 8.2 and just keep going. All right, 8.2, genotype versus phenotype. A genotype is an organism's genetic makeup. It's the genes that they have. So it's the alleles that the organism gets from their parents. For the purple flower color, big R is going to be purple, little r is going to be white. These are genotypes, is the two-letter code here. This purple and white are something completely different, and we're going to talk about that next. All right, so phenotype is the physical expression of the gene, what you see as you look at this organism, or as you look at this plant, or as you look at a protein, or whatever it is that this gene is coding for. So it's important to remember that dominant traits hide recessive ones. So in this kind of one example here, big P equals purple flowers, and they're on B's on this one. B's are a little easier to tell the difference between if they're capital or lowercase, but also big P little p is purple flowers. And the only time you get white flowers is if you get two recessives, little p. So even though these two are both purple flowers, they have the same phenotype, but not the same genotype. They have different genes, but they have the same exact look to them. So they're both the same phenotype. When we talk about chromosomes, we can talk about whether they're homozygous or heterozygous. And homo means same, and hetero means different. So same genes, different genes. Homozygous means both alleles of the pair are the same. Example, big P, or little p. Or homozygous here would be this one and this one. And then heterozygous, this is when the alleles in a pair are different. And they can be, in this example, would be little p, big P, little p. Or this one here, this heterozygous here. So they have different alleles on their chromosomes. They're heterozygous. If they have the same, they're homozygous. So a lot of genetics comes down to probability, and probability, simply put, is the likelihood that a specific event will occur. And then the equation for probability is this one here below. And so probability is the number of times an event happened versus the number of times an event could happen. And you'll want to have this equation, ready to do. You'll be doing some Punnett squares, and you'll be needing this equation when you do them. So the dominant trait happened 0.75, or 3 out of 4. It happened 3 times out of the 4 total crosses in Mendel's experiment. So 75%. If you want to times this by 100 and get a percent, you can definitely do that. All right, so predicting monohybrid crosses. So real quick, monohybrid crosses, we're only going to track one characteristic, and we're going to use this tool, this model called a Punnett square, which is a diagram that helps aiding the predicting of inherited traits. And if you've probably seen Punnett squares before, but if not, we're just going to go through a couple examples, get you guys used to using them, get you comfortable using them. All right, so our example 1, we're going to do homozygous versus homozygous versus homozygous, and we're looking at flower color here. So let's go homozygous purple, big dominant colors, and then homozygous recessive. And what you do with a Punnett square is you're going to take the big B all the way down, and then this big B all the way down, and then you'll take the little b across. So little b across, little b across. Then they'll take this one across, little b across, little b across. So now we have our genotypes. We need to talk about our phenotypes. The probability of purple, we got 4 out of 4 purple plants here. So this equals a probability of 1, or you can think of it as 100%. All of these things say the same thing. For white, we got 0 out of 4 equals 0 or 0%. So in this cross, we got no white. All of those offspring turned out to be purple in this cross. Let's look at a different one. All right, for homozygous versus heterozygous. Let's do big B. Oh, excuse me, big B, big B. And I'll do it in a different color this time to really emphasize this. We'll do big B, little b. So in this one, once again, you take this red big B down, and this red big B down. You take this blue big B over, and you take this little b, excuse me, little big B. You take this little b, and you take it over too. This one, now we have some heterozygotes, but we still have 4 out of 4 purple, or 1, or 100%, and 0 out of 4 white, or 0%. All right, let's go look at one that's a little bit more fun here. Heterozygous and heterozygous. So that means that there's a big B and a little b, and the other one also is big B and a little b. Now this is where they start to get fun. Big B down, little b down. I'm putting the little b in the second spot here. This is just you always want your recessives in the right hand side. It just kind of makes it easier to read. All right, then we're going to take this blue b over, and this little blue b over too. So now our purple ones are going to be these three, because they have a big B. So that's 3 out of 4 purple, or 75%, or 0.75. And then for white, it's going to be 1 out of 4 of these are white, or 0.25%. 0.25 or 25%. So heterozygous crosses are fun. You should always see this 3 to 1 ratio in heterozygous crossings. All right, so we can talk about 2 different ways to look at this data. We can look at genotypic ratio and phenotypic ratio. So genotypic ratio is the ratio of genotypes that occur. So in this one, what is our genotype ratio? Well, we got n equals 0 out of 4 is one of the options we could have had. We could have big N, little n, the heterozygote, and in this case we got 4 out of 4. And we could have had little n, but we didn't have any of those, so that's 0 out of 4 too. So these are the different genotypes that we could have gotten from this cross. For the phenotypes, we could have either gotten, let's say that this is green or yellow pods. We could have gotten green, which is the dominant. We got 4 of those. 4 out of 4, excuse me. Or we could have gotten yellow, which would have been little n, but we didn't get any. That was 0 out of 4. So even though green can be big N, or big N, little n, the phenotypes still look the same. So the phenotypic ratio and the genotypic ratio are 2 different ways that you could display this, and they're a little bit different from each other. One's more useful sometimes, and one's more useful the other times. So one thing that we can do now is we can do things like test crosses. If you don't know the genetics, you don't know the genotype of an individual, you can use a test cross, and you can cross with an individual that has the recessive trait. So you have, let's say you have a green pea pod and you want to know whether it's green or yellow. You would take that green one, it has at least one green, and you would cross it with something that was homozygous recessive. If this is big G, little g, you'll get 50% of the offspring yellow and 50% of the offspring green. On the other hand, if it's big G, you'll get a big G in each one of these, and you'll know that that individual has to be, all of the 4 of them have to be green. So if you do a test cross and you get a 0.5 phenotypic ratio, it means that it's a heterozygote. If you get a 1.0 phenotypic ratio, you know that it has to be homozygous dominant. All right, so incomplete dominance. Most of the traits we've looked at have been dominant traits. So the dominant trait completely covers the recessive one. And what that means is big P, little p, and big P both have the same color. They're both purple phenotypes. In incomplete dominance, this is where the phenotype of the heterozygote, or the one that's mixed, is intermediate between the dominant and the recessive trait. So for this example, we have the red homozygous one is white red, and the white homozygous one is homozygous recessive. So homozygous dominance, big R, homozygous recessive, little r. And then the pink is a mix between the two. It is a dominant trait and a recessive trait. And this is what incomplete dominance looks like. So let's do an example here where both of them are heterozygous for a trait. So these are going to be 2 pink flowers that we're going to be mixing together here. Same as any other Punnett square, you'll carry this big R down, carry this little r down, carry this big R across, carry this little r across. So now we need to know the probability of red. Well, how many red do we have? We have just 1 red flower, and it's this one. The big R, so 1 out of 4, 0.25. The probability pink, these two, let's use a different color. These two here are pink. They have that big R, little r. So the probability of that was 2 out of 4, or 0.5. And the remaining one has to be white. You have to use 100% all the time. So it's 1 out of 4, which is 0.25% probability there. That's how you do some incomplete ones there. All right, so in codominance, both alleles are equally expressed. So if you have an allele, there's more than one dominant, and they'll both be expressed. An example in the animal world is calico cats. So these get a little weird. We start to give them some different names. So let's say for our brown one, it's going to be B1. Our black one will be B2. And then if they're going to be a white cat, they'll be just little b. So let's take a heterozygous brown and a heterozygous black and cross them together. So that brown, B1 with a white, and then a black, B2 with a white. Okay, so we'll take this brown one down. Just gonna put them over here too. I'm gonna put the B1 there. Put the B2 there. And then I'm gonna write in white here, just in case we get any whites. This is when it starts to get a little hectic on Punnett squares. All right, we'll carry this little b down. We'll carry this black one over, B2, and we'll carry this white allele over the little b. Okay, so looking at this, the probability of brown. Now, brown is B1 or B1 and a little b. There's one of these here, so it's 1 out of 4. The probability of black, it looks the same kind of, but it has the B2 instead of that, 1 out of 4. The probability that has both black and brown is also 1 out of 4. And the probability is that white is 1 out of 4 as well. So all of these have the same probability in this particular cross. So a time we see this really commonly is in blood typing. So with blood typing, both A and B are dominant and type O is recessive. So a lot of times you'll see it written as IA for blood type A, IB for blood type B, and then little I is blood type O. So we're going to take a type O blood, and we're going to mix it with a type. That's not a very good color. Let's do a type A blood. But let's do an A blood that's a heterozygous for a recessive. So even though this has the O there too, it's going to be type A blood. I'm just going to real quick make sure these are all the same instead of, I'm not going to use the I's this time. You'll see the I's on an assignment. I just wanted to show them to you, but I'll use the A's and the O's now. All right, so for this one, excuse me. Bring this little O down. Bring this big A across. Bring this little O over. So the probability of A, there's two A's here, is 2 out of 4, or 0.5. There's no B's, and there's no AB's. There's two O's, though, so 2 out of 4, or also 0.5 there. Okay, now we said that A and B are codominant, so let's do one that has heterozygous for type A and heterozygous for type B. And do this cross here. So we get A down, O down, O down. We're going to get B over, BO, haha, O over, and OO. So for this one, how many type A's do we have? Well, we have this one here, that's AO. So that's 1 out of 4, or 0.5. How many type B's do we have? Well, we have 1. 1 out of 4, 0.5. For O's, we have also 1. Oh, 0.25 on all these, sorry. .25. And then that's going to be all the way down. And then for type AB, we only have 1 of those. So that's 1 out of 4 as well. So when you get these codominants here, it starts to get a little tricky for your Punnett squares. All right, the next type, we're going to take type O and cross it with type AB. We'll bring the O's down, bring the A over, bring the B over. So we have 2 out of 4 A's, or 0.5, and 2 out of 4 B's, or 0.5 again, and then 0 for the rest of these guys. So, couple sample monohybrid crosses with this one's with codominance, but a lot of the ones that you'll start out with don't have any codominance. They'll just be pretty simple, which is good.